Hello all. In food and dairy microbiology, today we are going to discuss on the topic spoilage of seafood, milk and milk products. Seafood. Seafood is highly perishable, especially fish is highly susceptible to contamination and spoilage compared to other seafoods. Contamination. The flora of living fish depends on the microbial content of the waters in which they live. Usually, the fish contains normal flora on their skin, like Pseudomonas, Acinetobacter, Moraxella, Flavobacterium, Cornybacterium, Vibrio, Bacillus, etc. The bacteria on fish from northern waters are mostly psychrophiles, which means they live in very low temperature, whereas fish from tropical waters carry more mesophiles. They live in optimum temperature. Freshwater fish carry freshwater bacteria, which include members of the most genera found in salt water plus species of Aromonas, Lactobacillus, Brevibacterium, Alkaligenes, and Streptococcus. Other than this, intestines of fish from both sources like the seawater source and the freshwater source contains bacteria of the genera Alkaligenes, Pseudomonas, Flavobacterium, Vibrio, Bacillus, Clostridium, and Escherichia. Boats, boxes, bins, fish houses, and fishes become heavily contaminated with this bacteria. As said in this point, the fishes become heavily contaminated with this bacteria because the bacteria will be present in the contaminated fish. Oyster and other shellfish contain alkali genes, Flavobacterium, Moraxella, Acinetobacter, etc. These fish thresh water bodies and pick up soil and microorganisms. Whenever the water comes in and go out of the fish's body, the soil will enter into the fish which contains microbial flora that will contaminate the fish. Number of microorganisms on the skin of fish can be influenced by the method of catching. The catching methods are enormous, like using nets, using hands and all those things, and using many other ways. These methods influence the contamination of the fish. The bacterial content may be present more in the soil, that means in the sediment. Whenever the fish nets are trawled for a long period at the bottom, it may lead to the bacterial contamination of the fish. Fish cakes and fish sticks may have contamination from potatoes, spices, and flavorings, etc. So, preservation is very important when it comes to fish because fish is most susceptible to autolysis, oxidation, and hydrolysis of fats, etc. Fish contains more fats which is hydrolyzed easily, that will lead to easier spoilage, and its preservation is important. So there are various methods of preservation like evisceration, rigor mortis, use of heat, use of low temperatures, chilling and freezing, irradiation, drying, use of preservatives, antioxidants, preservatives incorporated in ices. Evisceration is nothing but gutting out all those internal organs, because whenever the fish is dead and it is catched, when we doesn't remove the internal organs like intestine, it may lead to easier spoilage and faster spoilage. So evisceration is an important process which should be done at the proper rigor mortis stage. Rigor mortis is nothing but stiffening of muscles after death. So maintaining the correct data on rigor mortis helps to prevent the fish from getting spoiled easily. And use of heat, use of low temperatures like chilling and freezing, and irradiation, drying in the sun, and all those things are the preservative methods of fish. And factors influencing kind and rate of spoilage: the kind of fish, condition of the fish when caught, the kind and extent of contamination of the fish flesh with bacteria, temperature, use of an antibiotic, ice or dip. The kind of fish means the different types of fish, and the condition when caught includes the habitat and the condition of their death and all those things, and the kind and extent of contamination of the fish flesh with bacteria. When we discussed about the trawling of nets in the sediments, likewise, when we keep the net in the sediments for higher time, then the microbial flora in the soil may contaminate the fish. So, the kind and extent of contamination of the fish flesh with bacteria influence the kind rate of spoilage, and temperature and use of an antibiotic, ice or dip. There are many evidences of spoilage for the fish. The change is gradual from a fresh condition to staleness and then to inedibility. It is difficult to determine or agree on the first appearance of spoilage because the fish is normally have a bad odor. It is very difficult to differentiate its normal odor with the spoiled odor. So there are many ways to analyze the evidences of spoilage. The practical tests include trimethylamine tests used on salt water fish and other methods such as an estimate of volatile acids or volatile bases or a test of pH, hydrogen sulfide, ammonia, etc. or the methods. And Rea and Swan in 1949 described the succession of external changes like color changes, slime increases, change in ice, change in texture of the flesh, odor change, etc. In bacteria causing spoilage, at chilling temperatures like psychrophilic bacteria are Pseudomonas acromobacter flavobacterium at ordinary atmospheric temperature, that means mesophiles, Escherichia, Proteus, Serratia, etc. at higher temperature, thermophiles, Micrococcus and Bacillus. And the discoloration of the fish is a very important factor to check the evidence of spoilage. Yellow to greenish is caused by Pseudomonas fluorescens, yellow Micrococcus and others, red or pink by Sarcina, Micrococcus, etc., chocolate brown by Aspergillus yeast. Spoilage of special kind of fish and seafoods. Other than normal fishes, how other kind of seafoods are spoiled? Salt fish is especially spoiled by halophilic bacteria like Serratia, Micrococcus, Pseudomonas, etc. These bacteria cause discolorations. A red color is being common indication of spoilage. In moles are the chief spoilage organisms on smoked fish. Marinated or sour picked fish should be present on no spoilage problems unless the acid content is low enough to permit growth of lactic acid bacteria or the entrant of air permits mold growth. Whenever the marinated fish is uh, in the condition to permit growth of lactic acid bacteria, it may lead to contamination. And in chilled shrimps, Acinetobacter, Moraxella, and Vibrio, and crab meat, Pseudomonas, Acinetobacter, and Proteus. Raw lobsters is spoiled by Flavobacterium, Pseudomonas, alkali genes, etc. Crabs and oysters may contain species of Vibrio including Vibrio parahemolyticus. At temperatures near freezing, Pseudomonas, Acinetobacter and Moraxella, as we saw earlier, like the psychrophilic organisms cause spoilage, but Flavobacterium and Micrococcus species also may grow at some conditions. The spoilage is termed souring, although the changes are chiefly proteolytic. The protein in the fish is lysed, and at higher temperatures, the souring may be the result of fermentation of the sugars by usually the coliform bacteria like Streptococci, Lactobacilli, and yeast, etc. Early growth of Serratia, Pseudomonas, Proteus, and Clostridium may take place. An uncommon type of spoilage by Aspergillus yeast causes pink oysters. Next, we are going to see about milk and milk products. Composition of milk. Milk is an excellent culture medium for many kind of microorganisms. We all know that whenever we keep milk in the normal temperature, it gets spoiled easily because it's a very great source of microorganisms for their growth. It's a very excellent culture medium because it contains moisture, nearly neutral pH, and rich in microbial food. A plentiful supply of food for energy is present in the form of milk sugar, lactose, butterfat, citrate, and nitrogenous compounds in many forms like proteins, amino acids, ammonia, urea, and other compounds. And the accessory foods and minerals required by microorganisms are available. Some inhibitory substances like lactoperoxidase and agglutinase present in fresh milk soon gets ineffective. Ineffective in the sense whenever the milk is not kept in the particular condition, the inhibitory substances like lactoperoxidase and agglutinase also gets ineffective from preventing the spoilage. The presence of fermentable sugar and acid fermentation with bacteria is most likely to occur under ordinary conditions in raw milk. Where does contamination occur? In the case of milk, on the farm, whenever the milk is obtained in the farm, it uh, unless it is properly handled, it will get contaminated. In transit and at the manufacturing level, whenever it is transported, it should be transported in a sterile container with a particular temperature so that it doesn't get spoiled easily. And in the manufacturing level of dairy products, it should be properly maintained. Preservation. Preservation is by asepsis. Asepsis is nothing but keeping out the microorganism. Removal of microorganisms, the use of heat, pasteurization, ultra pasteurization, and steam under pressure. Use of low temperatures like refrigerated storage, freezing. Then the milk is dried. In the case of milk, skimmed milk powder and all those things, and lactic lactose powders for babies. Use of preservatives added and developed. Spoilage. Spoilage of milk is mainly by gas production, proteolysis, ropiness, changes in milk fat, alkali production, flavor changes, color changes, spoilage of milk at different temperature. As we said earlier, temperature plays a major role in preventing the milk from getting spoiled easily. Then milk also contains protein, so the protein will be lice if it is not properly stored. And the ropiness is nothing but the milk gets coagulated, then changes in milk fat. It may leads to the lysis of fat which will spoil the milk, alkali production, and the flavor and color is changed whenever the milk gets spoiled. Gas production is due to the gas producing microorganisms whenever they get a condition to grow on the milk. Spoilage. Flavor defects in cream. Cheesiness caused by lactobacilli. Rancidity resulting from lipolytic bacteria like the bacteria which lyses the fat and molds, and perhaps by lipase in the cream. Barney flavor will be produced by the species of Enterobacter. Malty flavor by Streptococcus lactis var malti genes. Yeasty flavor by yeasts. Metallic flavors caused by dissolved metals in highly acid cream. Highly acid flavor when the cream has excessive acidity, and flat flavor resulting from the destruction of diacetyl by bacteria like some of the Pseudomonas species. These are the flavor defects occurs in cream and milk whenever it gets spoiled, and the mentioned organisms are the causes for the spoilage. Then flavor defects in butter. Fishiness caused by Aeromonas hydrophylla, ester-like flavors resulting from action of P. fragi, skunk-like flavors caused by P. meptica, Roquefort-like flavors produced by molds. The Roquefort, like Penicillium roqueforti and Penicillium camemberti, are the organisms is used in cheese production, and these organisms, when it is not properly used, it may also lead to contamination and produce molds. The reference is from Food Microbiology 5th edition book by Frazier and the below given link. Thank you.